Hello. So in this video we are going to go over enzymes. This will supplement what was given to you earlier in Enzyme Basics, but please note that there are some advantages to video lessons. The power of video lessons is you can pause them whenever you feel like you need to go to the bathroom, eat something, or you lost your train of thought. You can rewind them. Same reason. And redo them when you think that you may have forgotten things. So in this lesson, we will, let me keep on moving myself out of the way. I seem to be in the way often. But we will describe the importance of enzymes. We will discuss models to represent how enzymes work, and we will demonstrate factors that affect the rate of enzymes. But to begin with, I'd like to show you a phenomena. So in here, pieces of potato. And I want to show you what can happen when you mix potato with hydrogen peroxide. Now, I am placing that piece of potato in here. It is wet because it is recently washed. And let me show you what happens when just ordinary potato gets mixed with hydrogen peroxide. I know you see the bubbles. All right, that's an ordinary piece of potato. Let me show you some alternatives, and we'll consider that a control. That's what normally happens. I have potato here that has been cooked. So quite a bit of heat has been added to it. I'll put that in another test tube right here, and I will add the same hydrogen peroxide. That's H2O2, and we will see what happens to this. And doesn't look like much now, does it, in comparison to the control? The control is the one I'm wiggling now. All right, and we'll put in a third. This one was mixed with acid. And the hydrogen peroxide. And there it is. Now I'll let them sit as I prove to you what acids are. I'll just put this aside. And if you recall from earlier classes, I showed you this pH paper. It's an indicator. It has chemicals inside this paper that will change color when acids are mixed with it, or bases, or, well, bottom line, it's going to measure the pH level. And I'm going to go ahead and rip off a piece of this. Now I will show you. This is hydrochloric acid. I'll get it a little bit wet with that. And you see, according to the chart, it is very, very low, extremely acidic. And the opposite of an acid is a base. I'll put it in another location. And you can see, according to the scale, it is extremely high. Halfway between is a pH of 7, which represents what water is. And what I put in here was acid. Let's take a look at those samples again. Here is your control. You still see the bubbles. Here is the one that was boiled. No bubbles at all. And here is the acid. Also, no bubbles. So what happened? Why are they so remarkably different? Well, what's at work here is something called enzymes. And interestingly enough, your body makes this substance, let's get a pen activated, called H2O2. That's hydrogen peroxide. Now, it's poisonous. It will kill you. Fortunately, your liver makes a substance called catalase. And this is an enzyme. It ends in ASE. Just about every enzyme we know of is intentionally put down with the ending ASE. And what this will do is it will convert the H2O2. We'll put an arrow here to say that it's becoming something new. And it will become H2O plus O2. O2 is in our environmental conditions is in gas form. So this is a liquid. This is a liquid. Hey, guess what those bubbles are? You guessed it. They're the oxygen. All right, so now, why is it that a potato would create those bubbles? When I mentioned it's our liver, well, think about evolution. We all have DNA. If you go back far enough, we all have a common origin. Turns out, potatoes make catalase also. And we can demonstrate how it is temperature and pH affects enzyme activity. So, quickly, let's do a little bit of a review. Which one of these organelles makes proteins? Well, it would be one. Those are the ribosomes. Make that a good looking R. Okay, which one permits passage of raw materials for protein synthesis? Well, that would be 4. That's our cell membrane. We have to get materials in there. And which one has the instructions for the protein synthesis? Well, that would be our nucleus. And it has DNA inside of it. The instructions for life. So I'm just going to put this up there rather quickly. Run through it fast, because you had another video which said many of the same things. So enzymes are specific types of proteins. They speed up chemical reactions. That means that they are biological catalysts. Breaking down this word, biological relates to living things, and catalyst is anything that speeds up a chemical reaction. And they are very specific to the substrates they work on. Roll back to what we have done in class. If this represents a protein, each individual piece here is an amino acid. So you start linking these together. That's what the ribosomes do. And then eventually, if you zoom out, as in Google Earth, you'll notice that they start taking shapes like this. All right, now that you have these shapes, you start zooming out further, and you'll notice that the shapes get even more complicated. And in fact, because of the shape, this is how enzymes work. You see, they end up with specific locations where other molecules that need to react can land. And it helps them out. It makes them go faster. Moving right along, there's a quick analogy I like to use. And that's, what if my wife and I were at the same location and we were back to back with one another? We could have gone the entire night without ever meeting. Well, turns out her friend was there and got to know me briefly from conversation and thought that we might find each other interesting. And she tapped her on the shoulder. We faced each other. We talked. It worked out. And now we're a couple. Conversely, an enzyme could also help you break apart like a divorce attorney. It just makes it happen faster. Now, this little illustration shows you an enzyme, and if this represents the protein known as an enzyme, this right here is called an active site where the magic happens. And right here we have a starch molecule, and it's going to fit right in. See, the shape matters. See how it locked in? And in this particular case, after it locks in, it cuts it, and we end up having, get this out of the way, and we end up having multiple pieces. So let me show you this briefly. This is a substrate, the chemical in which the enzyme is going to act upon. This here is the enzyme, and this is the active site. Okay, here's our substrate and enzyme together, the enzyme substrate complex, and now the substrate has become 2 pieces. This shows it also. You can pause it and run through it slowly on your own. I am going to continue and show you what happens when an enzyme, well, gets out of shape. And if an enzyme gets out of shape, we got a problem. That active site is no longer available. So quickly show you this, and to show you this, this enzyme's job is to break down substrate, predict what shape the substrate might have to be to fit in snugly. Let me drag in for you the answer. Okay, here we go. And if you thought square would fit into the active site, you're absolutely right. But what happens if the environment changes and that enzyme now changes shape? Let me show you. Oh no, liar. Here we go, I think. It worked earlier. All right, I'll show you another way. That enzyme would go from its normal shape to this. And if it's in that shape, that substrate, which is a rectangle, I said square earlier, a rectangle, will not fit in. And now it's not doing its job. So we have ourselves a broken enzyme. Let me show you these now. To get these definitions down, what I ask that you do is allow me to scroll through it, but you pause it when you see necessary to take notes. So I'm going to scroll through it rather quickly. Again, you're pausing as you need to. And onward. So we have this thing called pH. I showed you the I showed you this thing to demonstrate that it does have values. I showed you that we do have acids and bases and how it affects the enzyme's ability to work with use of potatoes. So this one was the acid. Again, still not working. Temperature. What does temperature do? Well, when the temperature is too low, particles don't move so fast. The enzyme cannot find the substrate, so it's not going to work so well. But when the temperature gets too high, like what I did to the potato, I boiled that thing. Well, the enzyme gets broken. And here it is. It's still not working. And again, let me show you the control. It's still working. Most definitely still working. Look at those bubbles. Oxygen's being produced. All right. So temperature will affect the rate of enzyme activity, pH will, and, well, let me see. Nah, I think that will do it. I just want to ask you one last question to finish this up. And we kind of started with this, but it's a little different now. From the same picture as before, which organelle synthesizes enzymes? Again, it's one. If enzymes are proteins, ribosomes are still the answer. Which organelle permits the passage of raw materials for enzyme synthesis? Well, if enzymes are proteins, we still need the cell membrane to allow the amino acids in so we can build them. And which organelle has the instructions for enzyme synthesis? That's still the nucleus, because DNA leads to the production of proteins, and enzymes are proteins. So, rolling back to our objectives, we will describe the importance of enzymes. Done. Chemical reactions require them. We will use models to represent the enzymes. Models, many illustrations. Done. And we will demonstrate the factors that affect enzyme reactions. Done. I hope this helped.